from Microbe TV. This is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld.
Hi, Vincent. How are you today?
I'm well, thank you. And yourself?
Let's not talk about it. Let's just.
Are you fair to middling? So tonight, Amy has a ribbed neck cashmere blue sweater, right?
Turtleneck. Yes.
It's got ribs. It's got ribs.
Yes.
Yeah. And I have a what is this, lime green?
Pistachio.
But I don't think
pistachio.
Anyone cares. Is it pistachio? Oh, good. That's a great color.
Of course it's pistachio. Of course it's a great color. I picked it out.
We looked at the catalog and Amy said you should get this. Yeah?
Yes. Especially in the summer. It's a good color with a tan, with an olivey complexion tan.
Okay, let's start in with
my big news today.
What's your big news? Oh, yes, you could talk about your big news. Go ahead.
You don't even know what my big news is.
Well, that's why I said you can talk about it, right?
Yeah. So my painting is coming on Monday.
What painting?
My EV68 painting by Jen and Animal Party is coming on Monday.
Oh, that's cool. Awesome.
I'm very excited. Hopefully she'll be able to come after 1:30, after I come back from you. But yeah, very exciting. It's my big news, my big painting.
This is Jen from Rhode Island, right?
Yeah, the nurse.
Wait, she's bringing it to you?
Yeah.
Oh, that's very cool.
Yeah.
Very good. Tomorrow?
Monday.
Yeah, I won't see her.
After lunch.
Oh, you have to go back to Columbia and then you're going to see her there?
Yeah.
Okay.
What do you think? I'm schlepping my painting around so it gets broken on the subway?
No. Nope.
Schlepping my painting around so it gets broken on the subway.
All right, our first question is, hello, in Malaysia, Pfizer vaccine for 5 to 11 years is given 8 weeks apart. How is it in the U.S. and any study for this?
Yeah, there have been studies about spreading out the doses. And in the U.S., it's not done well because it's still done every 3 weeks. Or 3 weeks apart.
So for 5 to 11, it's going to be 3 weeks or 4 weeks. Yeah, 3 weeks for Pfizer, right?
Mm-hmm.
Okay. Any thoughts on why or how some folks are asymptomatic with COVID whilst others suffer massive inflammatory response?
Genetics.
Genetics of the host, right?
Yep.
Are you saying that people are different?
Well, clearly.
I like the wide-eyed Amy look, which means, are you serious with that question? Yes?
Pretty much.
I want to thank our moderators tonight. We have Vanity Nutrition from what? A million miles up, 35,000
Oh, she's on a Bezos?
She's on a plane. Not on a plane, not a Bezos, yeah. We have Steph, and we have Tom. I think that's everyone. Did I miss anyone? Let me know. That's who I saw so far. And it's a nice little group tonight. We have 360 people. I would be happy with 360 people, wouldn't you, Amy?
Yeah, for sure.
I can't read the name because it's backward, but Vincent always asks, please hit the like. Where is it? I don't see it on my phone. So if someone can tell backwards name person.
It's Neil Ritchie.
Neil Ritchie. Okay, where the like button is on the phone. Okay. My Filipino relative had 2 jabs of Pfizer 4 weeks apart. She also had COVID. Would a booster protect her from Omicron if she had Delta?
Yes.
But isn't she all set with 2 doses and a recovery?
Yes.
Yeah, I don't think you need a booster.
She doesn't need a booster, but if she wanted to be like have mental confidence
Yeah.
that she was protected, she should get a booster. Science and mental confidence do not always equal each other.
And Les is here. I'm sorry, I forgot Les. Thank you, Les, for moderating. Everyone wants to know where the painting is. Show it to us, but you don't have it yet, right?
No, it's coming on Monday. Monday is Monday.
So
Today
next
is
Wednesday you
Wednesday.
can show it to us on the live stream?
Maybe. We see how big it is. If it's too big, no.
Oh, it can't be that big. How could you carry it to New York?
I don't know.
Well, maybe
Somebody
she's
sent
here
you
tonight.
71-pound thingamabobber, you know?
Yeah, he's usually here in the live stream. I will. I'll thank him when it comes up. Richard wants to know the New York Times on the new vaccine. This is the Sanofi, right?
Yes.
Efficacy of 100% against hospitalization, 75% against moderate disease, and 58% against symptomatic. Impressed that they spelled all that out. Is media improving? No, media's not improving. Probably the press release had that in it, and they just copied it because I don't trust. I don't feel that they're learning anything, and I'm sorry to be cynical, but I see day after day.
It was by the same lady that you.
I don't like.
Yes.
Have you ever had a mouse escape in the lab?
A mouse escape where?
You run around the lab.
No.
I did once. Can I tell the story?
Sure.
Very, very brief. So one of my students had caught a mouse in her apartment in New York City. And she said, "You want it?" I said, "Yeah, bring it in. We'll look for viruses." So she brought it in a box. I put it in the chemical fume hood in our lab, which you know, Amy, right in the corner.
Mm-hmm. Mm-hmm.
I cracked open the top and out it went. Gone. Zoom. So fast. Because I'm used to lab mice, which you open the top and they sit there. But this guy jumped out and he was gone.
Oh, wow.
But it was a wild mouse, so he's just returning to his milieu, right?
Yes.
The like button is at the bottom left at the end of the row of action icons. Okay, thank you, Pete, for letting us know. And yes, media is catching on to memory T and B cells. Right. Why does it take 2 years? They're journalists. They're supposed to be able to do this. What am I missing, Amy?
Well, it took 2 years for the scientists.
Well, we were talking about it a long time ago, weren't we?
Well, I was talking about it when I wrote my first SARS-CoV-2 grant. So, I don't know. I'm not the right person.
What is the Sanofi vaccine, Amy? What is the basis for
protein?
it?
It's protein.
Okay. And we're still waiting to hear about Novavax, right?
No, it put at the end of the article that I text you and Daniel that was in the Times by a Porva, it said that Novavax had applied for EUA at the end of January or something. Told you at the end of the article. Good to know that you read my texts.
Recent study suggested improved fomite transmission with Omicron. Codswallop or possible? Codswallop, I love it. I wouldn't believe it. I don't see why you would have more fomite transmission and how you could possibly know so quickly. What do you think, Amy?
Codswallop.
Codswallop. Do you know what that means?
Crap. Crap on a shingle.
Oh, there's an old Amyism. Crap on a shingle.
Crap on a shingle.
How covered is a person after only one Shingrix vaccine? Does it matter if said person missed the dose at the prescribed time? No, you can get it later. It's fine. But you should get the second dose because it's a 2-dose vaccine. I can't really say how well you are covered, but obviously they don't think it's enough to do it. That's why they recommend 2. Did you hear that 2 cases of early onset dementia have been reported in people with long COVID by UK scientists? Should we be worried?
I don't know. I didn't hear that.
I would say you don't know because maybe they would have developed long dementia anyway, right? Irrespective of long COVID. So I think you can't make conclusions yet about it. Yeah?
Just sounds like an association. Like I got run over when I got vaccinated.
Do you think it's possible for society to dig itself out of the vaccination misinformation hole that social media has dug?
No.
No?
No. Because it's social media just expanded it exponentially. The vaccine misinformation platform has been growing in the United States since the 60s and then really took off without social media by Andrew Wakefield. So no.
Can we make any difference, we at TWIV, Microbe TV?
Yeah, sure.
I mean, I think that's our way of dealing with it. We just tell people the right information, they spread it, and many people tell me, they spread it as much as they can, so I think that helps. That's the way I think to do it. Amy is my hero.
That's cute. Very good.
And welcome, Frank. Thank you for moderating. Appreciate it. After a positive home COVID test, the doctor said to wait 90 days before testing again. Could that be correct? Seems too long to wait for proof of negative. Why would you wait 90 days?
Because the doctor's then going to do a PCR. And it'll take at least 90 days. They want to make sure you're negative and not that it's persistence of RNA. And that seems to be when RNA persistence is no longer detectable.
These are just fragments of RNA, right?
Yeah. But, most people are stupid.
Did I understand in the last TWEVO that Omicron is approaching the definition of a new strain? No, that was... Nels brought up this opinion piece where they are suggesting that. And actually, we covered that on TWIV, which hasn't dropped yet. And you should wait. I don't think it should be a new strain or a serotype, in fact. I think Amy doesn't think so either, right?
I'm on the fence.
Okay. Everybody loves your sweater.
It's good.
Wow. Do we know what properties of poliovirus cause paralysis? Properties. I'm not sure how to answer that. What properties, right? The ability to enter the central nervous system, right? Neuroinvasion, and to reproduce in neurons.
Well, neuroinvasion is not a property that causes paralysis because mumps neuroinvades and is not neurotropic or neuro.
Yes, but you can't get away without it, though, right? I would say it's necessary, but not sufficient. Yeah. Anyway, but also reproducing in neural cells obviously is a big one, right, Amy?
Right, because you could just kill the neuron that's innervating that specific muscle, right? You could just go kill the axon of that. You don't have to go all the way into the spinal cord, which was my complaint about a paper, right?
Yes, indeed. All right, this is for Amy. Is cross-reacting immunity between cowpox and smallpox viruses attacking identical epitopes or similarly shaped epitopes?
Could be neither.
It could be either one or both or neither, right?
Right.
It could be an unrelated epitope that happens to bind the antibody, right?
Right.
Can you explain what a new strain means for the pandemic? I know what Amy's going to say. One word.
Shit show?
No, nothing. Oh, shit show. It means nothing.
You said one word, so...
Is that one word or is it two words? I thought that was two words.
One word. It's a compound word.
Okay. No, I don't think it means anything. We were actually discussing this on TWIV yesterday. What does it mean? It doesn't mean anything. Get vaccinated. The vaccines work. I
Although
don't
I
see.
was very surprised on Tweevo you let Nels talk about our values, which was a crock of shit.
Yeah, but I also railed against our values. Did you hear that part? Or did you just
I
shut it off
did.
when Vinny started talking? Huh?
I did. I heard. I just was like flabbergasted that this genius started talking about our values and couldn't figure out that it's a non-existent, ridiculous number that we've made
Hey,
up.
look, he's a genius. He has a genius award.
Well, he should give it back.
No, no, he is a very
Yeah,
politically
yeah.
correct guy.
I should have a genius award.
Well, I agree with that. Totally. You're a genius, for sure. I agree. I agree with that. But, these are politically influenced, as you know. I mean, who's
Fine. I don't need, I don't, I'm, you know what? I'm fine just being me. I think me is just fine.
All right, I don't know the answer to Mike's question. If methotrexate with folic acid have opposing action on immune stimulation and DNA replication, would high folic acid enhance vaccine response? High folic is linked to severe COVID. I have no idea of any of this. Do you, Amy?
I don't even understand the question, really, to be honest.
Well, methotrexate's an immunosuppressant, right?
Right. And
I mean,
folic acid is what? Has opposing. So folic acid is enhancing DNA replication? Is that what it says?
That's what he's saying, yeah.
So would high folic acid enhance vaccine response?
Not necessarily.
So why are you replicating the DNA?
Yeah. I don't see where it would do that, no.
And high folic acid linked to severe COVID-19, DNA replication has nothing to do with COVID-19. Is he linking it to like high DNA replication for enhanced inflammatory response? I don't think the cells are dividing. I think that they're just over translating the mRNA. It's a misregulation of translation, not a misregulation of DNA replication. And if even if you said it's a dysregulation of RNA synthesis, that's transcription, that's totally different set of enzymes than DNA replication. So it's not clear to me what the connection actually, why I care about what the effect is on DNA replication. I just don't get it. I'm not really clear.
Omar says you guys need NFTs. Now, I don't understand these non-fungible tokens, okay? Don't explain it now.
What is a non-fungible token?
It has something to do with, I don't know, current cryptocurrency or something. Somebody can explain it to me in an email. I don't get
well,
the
they can just send us. They can just send us the Bitcoin.
Actually, someone offered to send Bitcoin, but I don't know. I don't have a Bitcoin account, so I'd have to get one.
How hard could it be?
I'm sure it's not hard, but you know me, Amy.
Yeah, I know.
Do you know me?
I do.
For example, MZM sent me an email in regards to setting up a Discord. I know you did. I'm sorry I haven't gotten back to you. I will. It just takes time. I get lots of emails every day.
Call back in 2 to 3 months. Call back in 2 to 6 months.
No, not 2 to 6 months. I want to set this up in a month. I'll get on it.
I'm sorry. Hold a minute.
Are you sitting down?
Hello? These are not working properly. I heard something ridiculous about setting something up in an efficient time frame. These were not working properly.
You should be less disrespectful. I'm 69 years old after all.
Is that less disrespectful? It's just...
Hey, you got to admit, I don't look 69, right?
It has nothing to do with age.
Don't I have a boyish haircut?
Okay, sure.
Amy's about to say oi. Denmark has removed all of restrictions the UK about to likely outcomes with Omicron BA2. I think it's a good move. We have vaccines that work. No problem.
I think it's fine.
It's fine. You get a sniffles if you get infected with Omicron. All right. This is going to be a tough one, but I'm going to try it. If mice lie, monkeys exaggerate, and ferrets are not people, how do lab studies determine a virus's contagiousness and lethality?
Well, they don't. Lab studies have never shown virus contagiousness because that suggests transmission between animals, and that's really hard to do. And I don't think you could even show it for polio in the monkeys. I don't think you could show it in that it came from it was orally fecally transmitted between
No,
monkeys
no,
in the
you're right.
cage. So I don't know any lab study outside of the Fouchier and whatever the guy's name is in Wisconsin that looked at virus contagiousness. And lethality, I don't know what lethality, I don't understand what lethality means. You give X amount of virus and then you wait for the animal to die, right? So you can't extrapolate how lethal the virus is to
Yeah,
people
yeah,
because you have no idea what that infectious dose
yeah.
is. So
I mean,
I don't understand.
look at
I don't
the
understand the question.
look at the paper of Mike Diamond's that we did. Omicron is milder in hamsters. What does it mean? It still kills people, right?
Yeah,
So
and
it's
Daniel is very adamant about not saying that it is milder. And when they came out and said it was milder, they didn't account for the fact that 90% of the people have some kind of immune response to it. So if you were to just compare the numbers between Omicron and the first wave and the second wave, you're going to get a skewed result because that's when we didn't have any immunity. So I don't understand.
has Moshe stayed in touch with you about progress on MISC and super antigen motif? No, I haven't heard, but I could reach out to him, but no, he has not told me anything. Did you see UVA is going to do an ivermectin and fluvoxamine study? Yes. Yes. Looking forward to the results. For new viewers, can you explain what the Incubator project is?
Oh, it's a project?
The Incubator is a name that Amy came up with. Or no, wasn't it Anthony in Jersey City?
No, I came up with it.
Okay, he suggested something on the lines of factory, and you said Incubator. Right. It's the name for our studio, our recording studio on 7th Avenue in Manhattan. My daughter came in the other day, and she said to the doorman, "I'm going to the Incubator." And he said, "What?" He didn't know what it was. That's the Incubator. And it's supported by your donations, so we really appreciate it. Let's see. There was one here. Where was it? Amy, please describe the progress of the immune response over time to a SARS-CoV-2 infection. How long does the adaptive response take to develop antibodies? M and T. What's M? Memory and T cells? B cells? Okay. Go ahead, Amy.
So depending on what form of antibody you're talking about, right? So they think it takes like 7 to 10 days for you to get like an IgM or IgA response, and then it takes longer for IgG. And then it takes a couple of months for you to develop a memory B cell response, approximately 6 months or longer. And in that time frame, you're communicating with the T cells. Then they get trained, and then you probably after 6 to 8 months, you have memory B and T cells.
Thank you, James, for your contribution to the Incubator. And this, by the way, that's where we record all our podcasts. And we do everything there except this live stream. Although we have done a couple from there, right, Amy?
Yes.
Modding from the edge of space would impress. Indeed. Amy, what would you say is the most surprising detail mechanism about SARS-CoV-2 that was not expected?
Stupidity of the scientific community?
No, that's about the people, not the virus. They want to know about the virus. Okay, what one thing that you think was not expected? We answered this last week, actually, but you disagreed with me.
Well, I wouldn't say I didn't expect it. So just because people were not smart enough to investigate antigenic drift of coronaviruses doesn't mean that it never occurred, right?
I know, but yeah.
You just weren't smart enough to investigate it. You weren't interested, you didn't understand biology, and you made stupid conclusions or stupid assumptions. And I don't do those kind of things. Those aren't my way of thinking. My way of thinking is if all viruses undergo antigenic drift, show me a virus that does not. Therefore, my hypothesis would have been SARS-CoV-2 can undergo antigenic drift like anything else. So,
Does measles undergo antigenic drift? Measles
yes.
virus?
Yes, until it can no longer use the receptor. It's a seven base replacement, and that 8th base causes it to go extinct. Just like polio.
Amino acids.
Yeah, amino acids, just like polio.
Thank you, Lisa, for your contribution. Appreciate it. And here's Jen, Animal Party. The painting is looking good. I needed it to have a glow to represent your brilliance.
I like sparkle.
I'm
she
excited
sent me.
to
That's why she
deliver it.
made my snowflakes with sparkle, with glitter. Because who doesn't like a little glitter and a little sparkle? You got to have some sparkle.
Miguel says, genetics? Why siblings with supposedly similar genetics? They're not identical.
Nope.
They're never identical.
How
Your
could
cells
they possibly be identical?
Nope. So that's it, Miguel. They're different enough to cause these different reactions. No doubt about it. Thank you, Rob, for your contribution. Amy, is pistachio a fungus?
That's a nut.
It's a nut. Ay, ay, ay. And Rima is finally getting his smelling back after 2 years of infection. Wow. That's a long time. And here it is. The painting is 17 by 17. Not too big to show us next week, Amy.
Okay.
That's about the size of the HIV painting behind you.
Okay.
Okay. Take a picture of it. It appears to me that the focus on spike was too narrow in vaccine development, as I think you originally cautioned. Is that your view currently? I'm going to ask Amy.
I'm the one who pointed it out as being myopic.
Are you still of that thought?
Yes.
I had to say that paper we did where they have the 3 protein adenovector mucosal vaccine, that was very impressive. Don't you think?
Yes, I'm the one who sent it.
Yeah, of course. Am I getting any new nucleocapsid polymerase and spike? Are you getting any new virus models? No, I haven't gotten any new ones. I have a lot, and I have enough to show behind me at the incubator. So, but one day when I want a little diversion, I'll order a virus model. That's what I do. Amy, I think she likes to buy stuff, shoes maybe. I like to buy virus models. Would that be fair, Amy, to say?
I haven't bought shoes in over 2.5 years.
Oh, I'm so sorry.
So,
are shoes in your future, Amy?
It's unclear.
It's unclear. What's your favorite protein, viral protein, Amy? I have one.
Yeah, yours is RT.
How did you know? Did I tell you once?
Because I know.
What's yours? Helicase. No, just kidding. What's yours? I don't know what yours is. Keep it close to your chest. You keep it close to your chest. You don't share these things.
I'm not sure I have a favorite viral protein.
Okay. Apparently, Osterholm was on Rogan. This is what he gets for a scientist. Okay. Rogan tried to get weird.
Technically, he is a scientist.
Yeah, well, it's not the person I would have.
Well, I didn't say he was the right scientist, but you said this is he gets a, he it was
Yeah, I
like
didn't.
he wasn't a scientist.
No,
He's
he's a
actually a
scientist.
scientist. It's the wrong one, but he is a
Yeah,
scientist.
I
He
didn't say
fulfills
he
that
wasn't.
checkbox.
I didn't say he wasn't a scientist. All I said was this is the kind of scientist he gets. Okay? That's fine. It's good. At least he held his own. Priscilla says, when I first read about 1918 pandemic, I remember thinking how manipulable were people to allow the war to proceed when they knew it was spreading the flu. Oh my gosh. Are you kidding? War is like at the top of the list. That's what governments want to do.
It was at the end. It was at the end of
the war. Yeah. Would you be worried about travel if you were fully vaxxed but had Sjogren's disease?
Ask Daniel.
I really don't know. It's an immunodeficiency, right?
I think it's like lupus.
It's an
or
autoimmune disease. That's right.
Yeah.
Yeah, it's
I
an
think you have
autoimmune
to
disease.
ask Daniel.
Yeah, I don't know. You could send an email to daniel@microbe.tv. Yeah, and he will answer it. Spaghetti carbonara or linguine with clam sauce? Okay. Here's the thing, Taylor. Here in the U.S., you really can't get authentic spaghetti vongole because we don't have vongole, which are the kind of clams you can get in the Bay of Naples, or it's off the coast of Italy. You can't get them here, and that's what makes spaghetti with clams. Here we have regular Atlantic Ocean clams, and it's not the same. But if I could get spaghetti vongole, that would be my choice over carbonara. Carbonara is delicious, but it's a bit too. What's the word when you? It's too much.
Rich.
Like too rich,
It's
but there's
too
a better
rich.
too
Indulgent. Too heavy.
indulgent. Yeah, I can't think of the word. When you buy too many diamonds, it's too rich. Do you like carbonara?
I don't think I've ever had it because I don't eat pancetta.
The media
And
is
I'm not a
making
big egg fan. I don't really like
the
eggs.
media is making BA2 the next boogeyman. Should we be concerned?
Why is the
Amy?
boogeyman coming out of your closet? Are you concerned that are they concerned that the boogeyman comes out of the closet? If not, no.
The press just needs to make noise to sell their
Why?
product.
There's a whole war on.
What war?
Let's focus on the Ukrainian invasion. There's a war. So we don't need to, and there's a case of polio in Malawi and various other things. So we don't need to make up stories. We have enough to fill our plate.
The thing is, Jerome, vaccines work against all the variants. There's nothing to what the press says about BA2. They know nothing that Amy doesn't know. That's really the way I should put it, right?
I don't know. They know less than I do, that's for sure. Because I didn't see them in graduate school next to me with the yeast and the pipettes.
What's the criteria for when a coronavirus variant becomes BA1 versus 2 or the next Greek letter or SARS-CoV-3? I'm planning on taking the virology class this summer.
Well, SARS-CoV-3 is a completely different virus.
Yeah, it's going to be a different spillover from a different animal. Totally different, yes. Because just like 2 is very different from 1. But variants are named by how diverged they are from the pre-existing, the previous viruses. And it's done based on sequence of the genome. You should read the commentary on serotypes. What is the name of it, Amy? You sent it to me. Should Omicron be a new serotype? That was the name of it, I think.
Yeah, it's in Nature Review Microbiology.
It talks about how you name a new variant or how you decide that there's a new variant. Of course, a variant of concern has many changes compared to the previous one. That's the WHO says. Yeah, Omicron has 54 changes. It's a variant of concern.
So it's just the number of changes, not the function of the changes.
I think it's the number, and also if they're in places that we think impact receptor binding and antibody binding. Yeah.
Because you could have 54 changes throughout the genome and 3 in spike.
No, totally right. It's got to be in places of concern in the genome. How's that?
That's better.
This is Ian who sent the box.
Yeah.
So what was in the box, Amy? She said it was really heavy.
Oliver told me it's a battery so that you could have continuous power for your servers. And I wondered where the server was.
Is that right, Ian? Is that what it is? A battery?
That's what Oliver told me.
I thought it was something else.
That's what Oliver told me. It was a battery pack.
Okay.
But 71 pounds.
71? Is that why it was sitting on the 14th floor?
No, it was sitting on the 14th floor because FedEx doesn't know how to read and pushes buttons and just drops off packages irrelevantly of the fact that it says 1-3 and not 4-1-4. But it's why I didn't go pick it up yesterday. I waited for Oliver. Well, luckily Oliver came.
Is it known why unvaccinated people are more transmissible than fully vaccinated when infected? Does it have to do with shedding? Yes. Vaccinated people shed less. Isn't that right, Amy?
I'm not sure that an unvaccinated person who is on their second infection sheds less.
Oh, no, I agree. First infection.
I mean, it sheds more than a vaccinated person.
I agree with that.
I'm not clear that that's true. That doesn't make any biological sense.
No, but the first infection, you're going to shed more.
Yeah, the first infection, yes.
That's what we're talking
about. If you are unvaccinated and then reinfected, I would anticipate that you shed much less and much more similarly to somebody who's vaccinated.
Are there any RNA tertiary structures within the genome of SARS-CoV-2?
Yes.
Okay, there are. Why doesn't the malaria vaccine perform better? You're going to have to ask Daniel Griffin and Dixon Despommier that. Sorry, this is a virus show. Thank you, Dan, for your contribution to the Incubator. Really appreciate it. Amy, thanks for the recent paper and participation on Twitter. Super interesting and great to see a model of responding to colleagues asking questions like reviewers, responding with enthusiasm like peers.
Good.
What do you think? Okay.
It's good.
Polio in Malawi, right. It was type 1 imported from Pakistan, I think, correct?
Yes.
And why does it show up? Because vaccination levels drop, right?
Yes.
Do you know what Malawi is known for, Amy?
No.
Lake Malawi, which is slightly saline and has a certain kind of fish called cichlids. And
the Dead Sea is super salty, super saline. So.
And there's no fish in it, are there? Are there fish in the Dead Sea?
No, it's super. It has so much salt that you float.
Have you been? Did you swim in the Dead Sea?
I've never been. My sisters went.
What do you think about the Hep A vaccine? Is it worth getting as an adult?
Yes.
Well, it's a fine vaccine, but I only get it if you're going to places where there is Hep A. Have you had it, Amy?
Yes.
I haven't had it. I was offered it when I went to Brazil, but I declined it.
I believe I was vaccinated. I believe I had to get it to go to college.
It was once said that Colonel Sanders had nightmares that God was in the form of a chicken. Would a virologist's fear be that God is a mouse? That's a good analogy. I don't have such fears. Do you have fears, Amy?
I have fears, but not that fear. Yes. But I'm not a big proponent of gratuitous killing of mice.
Oh, no, we don't like to gratuitously kill mice, right? No. How about reviewing efficacy of inactivated virus vaccines papers instead of the usual mRNA? Well, Raz, you apparently have not listened because we don't do the usual mRNA. We have done inactivated vaccine reviews in the past. But so your tone is such that You're accusing us when in fact we've done it. And I'm happy to do it when new data come out, for sure. Is marketing of antibodies partly responsible for the lack of T-cell, B-cell recognition? That's an interesting
Marketing of
one.
antibodies? You mean from like Regeneron and the Skip Virgin's company?
Well, just pushing them. Like antibodies are the thing, right?
No,
You don't think so?
no. Comes from lazy scientists who started off assessing antibodies at the very beginning of the pandemic.
Okay.
Because it's easy. You do an ELISA, you do a neutralization assay, and then if you're smart, you can then parlay it into an antigen capture tech device or assay that you can then market to use as a diagnostic tool, right? It has nothing to do with pharmaceuticals. That's what Cromer did. That's exactly what Cromer did. Is it was easy, and then he marketed it, and he was smart, and he marketed it as he could make a lateral flow assay. Nothing to do with anything else.
Herpes linked to Alzheimer's. If SARS-2 is reactivating herpes, is that a risk for Alzheimer progression?
So on herpes. And first of all, it's not reactivating herpes. It reactivates zoster.
Right.
Totally different virus.
What are your thoughts on the Queen and her Covet?
She's vaccinated. She's old. Of course she's going to get Covet. But she should be fine.
Were you identified as gifted at an early age?
No. My sister was.
Only now that we're displaying Amy to the world do we realize that she's gifted, yes. But Amy is a retiring person. She likes to keep things to herself, right?
Yes.
She likes to
I'm
just do her work.
introverted.
She likes to do her work.
I'm very introverted. All right, it's 9:12 and I have Gila's that I need to take care of. So like 2 more questions.
Thank you, Pamela, for your contribution. Great appreciation for it. And here's some hearts for Amy.
That's cute.
Ian,
What is I befriended a chicken? So
I
this
don't know.
Sally Broker said I befriended a chicken I could not eat, but somebody else did. So she had a pet chicken? Like Howie? Who
Some
has a
people
pet
have
chicken
pet
running
chickens.
around their house? Besides
Ian.
Howie.
Ian, thank you for your contribution. So Ian says that it's he said in another email that's further down, it is a solar generator. It needs solar panel. You need a solar panel to attach to that thing that you got, and then it will power appliances. Thank you, Ian.
Well, I guess we should take it home then. Because I don't know how you're going to get solar into your incubator with all the windows boarded up.
Well, we have one window in the other room.
a little flaw in the plan.
Oh, by the way, Ian says, apparently birthdays are good for health. People who have more live longer. You agree, Amy?
Yeah, it's good.
Thank you for my favorite podcast. It offers discovery, adventure, and suspense. What more could anyone ask more? Well, that's a great summary. Thank you, Pamela. Really nice of you.
Good. All right.
And thank you, Michele, for your contribution. Okay, here's a question, Amy. If you two couldn't take Paxlovid, what would be your next choice of therapy?
I'm sure yours would be Molnupiravir.
Molnupiravir, yes.
Yeah, whatever.
What would you take?
Probably an antibody cocktail. I'm not a big believer in Molnupiravir. I think it was oversold tremendously.
Yeah, I would.
I
I
think
think I
it
would.
ranks right up there with remdesivir as a hoax.
Okay. Yeah, I agree with you. Can you do a TWIV on rabies? Yes, we will. I'll get a rabies person to come on. Do you know any rabies people, Amy?
No.
Okay. So TWIV 585, February 5th, 2020, VR. If this new coronavirus should stay with us and become endemic in humans, which is possible, people will forget about it, right? It will just be another viral illness. Yes. I think that's still true, don't you think, Amy?
I think it will be another viral illness. I'm not clear that people are going to, once it's endemic, that people are going to be so quick to forget about it. So are you talking 5 years after it becomes endemic, 10 years after it becomes endemic, or 100 years?
This was February 2020 that I said that.
Yeah, I'm aware.
I don't know what my time frame was, but I think 10, 20 years, yes.
No, more like 50 to 100. You need to have
Maybe.
all of these people die off.
What published statistic will we need to say the pandemic is over?
One case, less than one case per 100,000.
I just have 2 more questions. 3 more for you, okay?
Mm-hmm.
Polio has a selective affinity for upper motor neurons, does it not?
Upper?
Lower.
It's lower.
How about
The
EVD 68?
gastroenterologist. 68 is upper because it's respiratory. That's where it enters.
Yeah.
Polio is lower. Very few people were paralyzed in their arms.
Why did Amy decide to become a virologist? She met the most brilliant virologist of all time and said, I have to do this.
Yeah.
I'm kidding. I'm kidding.
I know. But it's kind of true because I was rotating in a lab and we were doing a yeast 2 hybrid with core of hepatitis C. And it was clear he wasn't a virologist. And I needed, if I was going to continue, since my first rotation was a disaster because the guy was an idiot, I needed a better, I needed to have a better virology background. That's how I came to you.
Okay, that's good.
Yeah.
They're like an old married couple. Who are they referring to here, Amy? Can't be us.
You and me. If so, I want to be in charge of the bank account.
You already are. You're in charge of the lab bank account.
Yeah, but that doesn't have any money in it.
Yeah, it's empty, right?
Exactly.
Would that be called a Pyrrhic victory?
Yeah, I guess. I want an account that has money in it.
All right. We'll work on that. You want to go now?
Yeah, was that the 3 questions?
Those are the 3 that I wanted you to see.
Yeah, I have to go. My HeLa cells are calling. I need to plate them out for a plaque assay tomorrow.
All right, everybody say goodnight to Amy.
I'm anxious for this result. We're looking at the C3. Are you excited? C3. Very exciting.
Okay. When are you going to know? Tomorrow? 2 days, right?
No. Tuesday.
All right.
Do the plaque assay and then make plates, then do the neutralization, then do the plaque assay, then Tuesday develop. C3. Super exciting.
Okay, Amy, thank you very much. See you tomorrow.
See you tomorrow.
Bye-bye. Okay, folks, stuck with me. I'm very sorry. Is it not true the significance of Omicron is giving herd immunity, surpassing the vaccination? Well, I think it's really too early to say, right? I think it's boosting people for sure, right? And it's doing it in a mild way, especially vaccinated people. So don't get me wrong. If you're unvaccinated and you get infected with Omicron, you can get severe disease. This idea that it's mild, ask Daniel Griffin how many patients he has in the hospital who are dying or very, very ill with Omicron. But if you're vaccinated, you have a mild disease, and I'm sure it's boosting you. But it is killing a lot of people also, so don't get that wrong. So what you're suggesting is possible, but I think we need to wait and see. Do you know what the current status is of the Wuhan lab, WUHAN, and what they're working on? I don't. I actually emailed Zhongli Shi, invited her onto Twiv, but didn't talk to her about what's going on. I don't know. Thank you, Pamela, again for your contribution. Really appreciate it. And I'm sorry to be slow picking up questions. There's a lot of chatter amongst you guys, which is great. It is great. Is there any cross-reactivity between OC43 and Covet or any of the common coronas? So there is some cross-reactivity at the level of T-cell epitopes. So that is the proteins that are recognized by T-cells, which kill infected cells. And whether it contributes to any protection is anybody's guess. All we know is that it exists, but whether it helps you or not is absolutely unclear. Thank you so much, Mr. Seaman. I don't really look 35, but I would say some people think I'm in my 50s. It doesn't. It's okay. I feel fine, right? I'm healthy. But Daniel Griffin said I have a good BMI, whereas he doesn't. And that's great because I don't exercise, but I do run around a lot, and I really moderate my food intake. So I hope to be around for 20 more years to be doing this with you guys. Does codon de-optimization mean end in sight for attenuated deactivated vaccines? Can we avoid epitope changes like RBD loss with beta-propiolactone, the inactivator? I think codon de-optimization is extremely promising, and we have to see the results, right? Because these are replication-competent viruses that don't cause disease in theory. So, yes, they could be the end of inactivated vaccines where you grow up virus and just, propiolactone or formaldehyde inactivate it. But we have to see. It's going to be some time before we know, right? What study, RCTs, randomized controlled trials or otherwise, are you most looking forward to regarding treatment? Well, I think let's see. I want to know the ivermectin fluvoxamine results because people have been talking about it for so many months now. I want to know what's going on there. But I'm anticipating in the future some trials of therapies for long COVID, right? I hope that comes out. But right now, I don't know of any. If rhinovirus were tracked like COVID, how many cases per day do you think there would be in the U.S.? A lot. A lot. Thousands of cases per day during the rhino season, which is, the winter, essentially. And I think that's the case for most of the viruses that are with us, influenza, rhinoviruses, all the respiratory viruses, parainfluenza, adenovirus, metapneumovirus. I think if we tested, we'd be stunned at how many infections there were in a season. Not necessarily. What month are we in? February. Yeah, we're in the rhinovirus season. We're in February. So put it in perspective. That's we see all these COVID cases because we're testing to a certain extent. And I'm not sure the numbers will ever get much farther from it. It'll go up and down because I suspect in the summer the cases will go down. But we're going to have lots in the winter all the time. Are there good data yet on correlates of immunity? Is it possible to ever have a test that predicts whether someone is well protected, since a simple antibody count doesn't correlate? Well, I don't think so. There's a division in the field. So many people feel that antibodies are the correlate of protection, but it depends what you're talking about by protection, right? If you're talking about protection against infection, then yes. If you have high antibody levels, which you have, say, after an infection or a vaccination, then you're protected against infection. But those levels always contract within 6 months or so. But then you're still protected against severe disease, and that is probably T cells. So you have to specify what you're talking about. When you talk about correlates, I think it's really important. But I've been told by people who study B cells that there's no proof that T cells are important. And I think that's probably correct, because we can't measure T cells as readily as we can antibodies by just doing a neutralization test. So if we could develop a better assay, we could get that. We could get more evidence for T cells, but we don't have it right now. It's a good question. Regarding the rabbit virus in Australia, is the mutated virus less virulent in rabbits in Europe? Is the original more virulent in rabbits in Australia? So, all right. So the idea was they took European rabbits and brought them to Australia. Devoid of predators, they multiplied to huge numbers, right? And so they introduced a myxoma virus to try and wipe them out. And within a year, both the virus and the rabbits had changed so that the lethality of the virus went from 99% to 30%. So is the mutated virus less virulent in rabbits in Europe? I'm not sure if that's been tested or not. Is the original more virulent in... Yes, the original is more virulent in rabbits in Australia. That's why it was brought there to begin with, so it would wipe out the rabbits. And in fact, in the first year, it killed 99% of them. But that 0.1%, man, they reproduced, and they were only killed at a rate of 30%, and plus the virus changed as well. Interesting story. So that's the best data, by the way, since you brought this up. Those are the best data that indicate that a virus can become less virulent in its host. Because we haven't seen it happen. We don't have good data for any other virus host system. Maybe transmissible gastroenteritis virus in pigs. And someone said, you didn't never talk about that. Yeah, I don't really talk about animal viruses very much in my course, but that's a good example. So this one, because it was an experiment, we had those data. Anyway, thank you, Samster, for your contribution, and thank you, Kent, for your contribution to science education at the Incubator. You should have a sign over the door that says Incubator. I do. And I printed one and laminated it, but recently I had some magnets made, and they're coming, and they're going to be stuck on the door. It says Microbe TV, Parasites Without Border, the Incubator, Suite 703, 7th and Manhattan. It's a 7th Avenue and 29th Street. It's just 2 blocks from Penn Station, which if you take a train to New York, you can walk 2 blocks and come visit. It's a cool place. Thank you, CCHH, for your contribution. Really appreciate it. And yes, please like the stream. Just give it a like. It's down below the video window, because we have 690 people here and 336 likes. So just move your finger down. It doesn't cost you anything. It just helps us to spread the word. So people say, how do you counter anti-vaccine, anti-science sentiment? By doing things like this, by having virologists like me and Amy talk, and the more people that hear about it, the better. Read something saying naturally conferred immunity from infection was found to create autoantibodies, which are allegedly harmful. Does this mean anything with respect to natural immunity versus vaccine? No, in fact, the best example is recently discovered link between infection with Epstein-Barr virus and multiple sclerosis, right? which we did on last week's TWIV. So you get certain people, when they get infected with Epstein-Barr virus, they make antibodies against the virus, and some of those antibodies cross-react with a protein in their nerve cells, and it causes multiple sclerosis. That's a natural infection. It is not a vaccine. So whether that would, we don't have an EBV vaccine, so I can't tell you if that happens. But, you know, in principle, it could, but I don't know of any example of it. When COVID hit, I was scared and sought out science instead of idiots on the news. I found YouTube, been watching ever since. Thank you, Ace. Really appreciate your having some confidence in us. And I mean, Amy has been right many, many times. And many of the things we said early on turns out to be right. You know, we're not saying we're right all the time, but we say question the data, wait till you have the data, and then it comes in and it shows, yeah, the virus wasn't more transmissible, yeah, it wasn't more virulent, et cetera, et cetera. What do you think of cheap homemade air purifiers? I don't think much of them. I just don't think you can purify enough air with a cheap purifier to make a difference in terms of viruses. Okay, Taylor, you're being a little cynical here. You say, so what use is a lab study in animals? It tells you principles. It doesn't make extrapolations to people. It doesn't prove anything in people. It gives you principles which you can then test or make observations on in people. And so that's why we do them. We do not do animal studies for the fun of doing them. That would be ridiculous. But you say, for example, in a mouse, let's say antibody, you find out that antibodies are the correlates of protection. And so then you can ask in people, is that the same or not? What exactly is being measured when COVID is said to be found? So you don't find COVID. COVID is the disease, okay? They find pieces of SARS-CoV-2 RNA, pieces of the viral genome, because they do PCR for a small fragment. So it's probably not infectious. Does it pose any risks downstream? No. I think it's zero risk. What it is, it tells you what's circulating in people, because you swallow your mucus, it comes out the other end, it goes into the sewage, and then we can tell what's infecting people. Or in the case of the recent episode, they found sequences that no people have. So the idea is maybe these are being excreted by animals, rats or some other animal that's being infected. Yeah. Your brain. How is your Luna? Are they vaccinated against SARS-CoV-2? Luna's fine. We have two dogs, Luna and Pepper, and Luna had a Guillain-Barré reaction to a vaccine, which she got last year, I believe. And so she had kind of paralysis for a week or so, and gave her steroids daily, and we fed her because it was hard for her to eat, and she recovered. But no, they're not vaccinated, no. So if it takes 6 months to have memory B cells, then is it fair to say reinfection is possible if the first wave of antibody declines in 3 months? Yeah, sure, absolutely. We start seeing reinfections after 3 months, after one dose for sure, or even 2 doses. So, but that's not a problem because you're protected against severe disease. Reinfection is not an issue. The press made it an issue. They said, we're getting reinfected. The vaccines don't work. Please go read an immunology book before you scare people to death. That's why you folks come here and you understand what's going on. Vaccines don't prevent infection. They prevent severe disease. That's how most of them work. What binds nucleocapsid protein to the RNA? And it's the same thing, non-covalent interactions, ionic, hydrophobic interactions between the protein and the RNA. They're not covalent interactions, but it's enough to, and the nucleocapsid proteins also interact with each other, so it makes a very stable protein-RNA complex. Is eradication of polio doomed to fail? No, I don't think so. I think what we see here is we have circulating vaccine-derived viruses, and when you let vaccination coverage drop, you can be infected with those or wild-type viruses in Pakistan and Afghanistan. So the first order of business is to finish type 1 wild-type polio in those countries. It can be done. It's just a matter of vaccinating, and it's hard to do that because there is terrorist activity in those countries. And then, once we get rid of type 1, then we have to switch to another vaccine, either an inactivated vaccine or these new modified attenuated vaccines that supposedly can't revert. I don't know if that's the case, that they actually cannot revert, but if they can't, that would be very nice. But I'm a little skeptical about that. So no, I don't think it's doomed at all. I think we've made really great progress, and we're gonna get to the end. Yeah. Can you explain why mRNA vaccines are vaccines and not gene therapy? Well, gene therapy is when you put in a gene to correct some defect that you have, and the gene becomes established in you, and it provides the wild type protein on a continuous basis so that your function is restored. So if you have a hemoglobin problem, you can give people the hemoglobin gene. This is an mRNA for a virus protein that lasts a couple of days and then is gone. The protein is gone, so it's not therapeutic. It's not staying. It's transient. So it's just an excuse that people give not to use it or to take it. So in no way is it a gene therapy at all. It's not altering any genes. It's quite clear. I'm not going to be able to answer this. Which living creature, to our best knowledge, is the most resilient to a wide variety of viruses? So by resilient, do you mean resistant to disease? That would be the most sensible qualification to your question, right? Because you could be infected, but if you don't get sick, it doesn't matter really, right? So, we haven't studied a lot of animals, so it's hard to know this. But I would say bats get a lot of infections, and they seem to be pretty resilient to disease. I mean, it's not that they don't get sick. They can get sick, and obviously some of them get sick from white nose fungus. But I think that's where I'd put my money right now. But they've been studied more than a lot of other wild animals in terms of virus infections. For triple vaxxed, how worried should we be about Omicron? No worries whatsoever. If you're triple vaxxed, Omicron is very mild, just like Delta is in every other variant. It's quite clear. That's what the data say. And if you've avoided SARS-CoV-2 so far, you'll probably encounter it at some point. You may get infected. You might get some sniffles, a little cold, like you do often, right? You get a cold. You get sore throat. You get sniffles for a couple of days. You don't think twice. That's what SARS-CoV-2 is going to be in an immune population. No worries. Now, I could say bad things so that you would keep coming to this live stream, but I would never do that. That's the fact. It's not in my DNA to tell you things that are not correct. How different are symptoms from variants and why? Yeah, carbonara is very rich. So here's the problem. And there's a great article in the New England Journal on why it's so hard to make conclusions about the virulence of variants by observational studies, where you just look at people who get sick. People said Delta was more virulent. And I could give you 10 papers that say that, and I can give you 10 that say it's not more virulent. because they're all done in different populations, different ages, different vaccine statuses, different comorbidities. It's just impossible to address it. So I have not seen any variant be more or less virulent. I think it may take a long time to get to that point where we have the data to do that. And you're going to need to combine it with some animal data. Despite Amy not agreeing that you can learn anything, I think you have to get some mechanistic data, not just whether it kills the animal or not, but what it's doing in terms of immune responses and in terms of reproducing in cells and so forth. And so it's a hard question to answer. And so why? We don't know, because there's no definitive answer on how different these viruses are, these variants are in terms of disease. Now, other people will tell you, of course there's difference. In my hospital, the sickest people had Delta. That's not data. I'm sorry. That's just anecdotal stuff that means nothing. Will Daniel be on this live stream? That's my plan. I would like to have him come on at, say, 7, and then, for an hour. And then Amy could come on at 8, and then I could wrap it up like I usually do. So we'll work on that. Yep. Not decadent for carbonara. Decadent? Maybe it's something like decadent, yeah. Yeah, so my stream here has every comment, even the ones that have been withdrawn. I have to be careful. So I have to look at them carefully and say, would this be a moderatable comment? And there's still a lot of those comments left here. I love that I tuned into this in the middle of a spaghetti debate. Look, I don't know if you're positive or negative about that, but we do, on Twiv, we diverge now and then because we're people. And we diverge here. If someone asks me about carbonara, yeah, I'll answer it. Why not? Is there any evidence of better cellular immunity for boosted compared to doubly vaccinated 4 months after the injection? So no, it's not relevant. So the boost, you're absolutely right. The boost improves humoral immunity, and it broadens it to neutralize Omicron, right? And so what it does to T cells, we don't know yet. Hopefully some studies are going to be done to address that. But I have a feeling it's not going to make any difference. I think the T cell response you get from 2 well-spaced doses is fine. We just don't have the data to address it. But you're absolutely right, and this has come up before. The boost looks good in terms of antibodies, but no one has looked yet. And now the information on the protection against severe disease now is coming in, right? And it looks like it's pretty good, but we still need to wait some more for that. Okay. If it takes 7+ days for antibodies to be produced, wouldn't replication of the virus be controlled by then? So antibodies are useless in new infection, and T cells is what stops progression of disease. Well, yes. By the time antibodies are being produced, virus titers are already going down, and so the antibodies don't play a role in clearing the infection. What they are doing is the T cells are helping to clear infection because those are being generated as well. But what the antibodies can do is limit the reinfection. So if you get reinfected, if you're in the 6-month window behind a vaccine where your serum antibody levels are high, then infection couldn't be blocked. But if you're beyond 6 to 8 months, you will get, if you expose the virus, it will begin to reproduce, and then you'll have a memory response in a few days. So you'll have replication. You may have a mild symptomatic disease, but you won't get severe disease. But again, it's not in 100% of the people. There are always outliers in the population because humans are genetically different, right? Absolutely. My brother and sister got hit by COVID hard. Is it possible I could get similarly hard? I'm immune compromised, so was wondering. All three of us are triple vaxxed. Well, if you're immune compromised, yes, you could be hit hard. But just because your siblings did doesn't mean that you would, because genetic differences among siblings can be big enough to make a difference in resistance to disease, for sure. And the fact that you're immune compromised, you have to be careful. Even though you're triple vaxxed, you may not have a great immune response. And so you should be careful. If you get sick, immediately, if you feel sick, immediately get tested, and you may be a candidate for monoclonal antibody therapy, which has to be done sooner rather than later, because your immune response may not be sufficient. No, we don't have any. We were toying with writing something about, what we've learned, and basically saying, like Amy says, we've learned that science can be a real problem in a pandemic, but I'm not sure we're going to do that. Gabriel, thank you for your super chat. Appreciate it. What other kinds of genomic changes could make a variant of concern apart from spike? Oh, that's a good question. Nobody knows anything about other genes, what they do, right? They're ignored. Only the spike is considered when they make a variant of concern, because you can do two things. You could see changes that affect the receptor, which to me are of dubious importance, because so what if a change makes the interaction more high affinity? How do you know it wasn't already good enough to get good infection? Maybe it's not going to make it any better. These are assumptions that are never tested with infectious virus. And second, you can see changes in the epitopes where the antibodies bind, and you can actually do neutralization assays and say, yeah, these changes affect antibody binding, so there could be of some consequence. But then people look at, after they're called variants of concern, people then look at the T-cell epitopes, people like Crotty and Sette, and they say, hey, most of them haven't changed. Everyone should be all right. But it's too late. They're a variant of concern. The press goes crazy. People go crazy. And in the end, get vaccinated. It'll protect you. I hate to keep saying that over and over, but nothing has changed, in my opinion. And you can have the epidemiologists screaming to the highest clouds as much as they want. It doesn't make any difference. Get vaccinated. No, the vaccines are not changing the natural selection. Any immunity changes the selection, or correctly should be applies selection pressure. And so that can be immunity from infection or vaccination. If you didn't vaccinate anyone and everyone got infected, they would have immune pressure, which would select for variants. So this idea that vaccines are doing something different from infection in terms of selection is pure BS. No basis in fact. Media keeps reporting that booster protection wanes considerably. Is this accurate, or is it something on a shingle, as Amy would say? This is BS. Booster protection. Yes, and first, I don't like the wane word. It's contract. The antibody levels contract. Don't they ever learn? Antibody levels always drop. Did you expect them to say high? No, you shouldn't, because that's never what happens after any immunization or infection. So they go down, and that's what they're expected to do. And you're going to get infected, but you're not going to get severe disease. I don't get this. What are they, manufacturing stories? Because I thought there were plenty of things to distract them for coverage. Can I do an overview of proper mask protocol? I can't. I don't have any masks right here. But Daniel Griffin did one on a TWIV several months ago. He showed with different kinds of masks how to put them on and take them off. And maybe one of the mods could find that episode and post it as a link here for you, Graham. I appreciate it. Why decline the vaccine? This is the hepatitis A virus vaccine. Okay, because you needed 2 doses, and I would be there after just the first dose. And so I was also not really going to the Amazon, which is where the risk was, so I didn't think I needed it. But if I could have gotten 2 doses, I would have, but I didn't want an incomplete course. How do people get parvovirus? From other people. You want to know the mechanism of transmission? There are some that are respiratory transmitted, and they can give you B19 in humans, can give you a rash. So respiratory transmission. But of course, they also affect your pets, right? Your dogs and cats. So you should vaccinate them against parvoviruses. Otherwise, they could die. Do you think repeat infections of COVID exhaust dendritic cells? No, I don't think that's correct at all. I think there were some anecdotal reports, isolated lab reports, but I don't think in the bigger picture that it is a problem. No. I just read John Barry's The Great Influenza. I was very glad I heard about early scientists here and on TWIV first. Might you invite him onto TWIV? Actually, we did. A long time ago, before the pandemic. Or maybe at the beginning, and he said he would come on, but I never followed up. So I'll contact him. I've been meaning to contact him. Yeah, I want to have him on. There are many people I want to have on. I need an assistant. I know that Daniel has interviewed some interns, and perhaps they could start helping me with these things. Okay, so here's Ian's gift. It's a Bluetti AC200 solar generator. It needs solar panels. It controls charging, has a battery for storage, and has an inverter so you can power appliances. Oh, cool. 70 pounds. Wow. Thank you, Ian. I'm not sure I can use it at the incubator. We only have one window. That's the problem, yeah. What are the risks getting vaccinated against smallpox as an adult? So the older smallpox vaccines had higher rates of side effects. But there are newer versions. Let's look it up, vaccine side effects. There are newer versions that are improved. And so, itching at the site of, because it's a scarification of the skin with the vaccine, so you're going to get itching, you're going to get swollen lymph nodes, a sore arm, fever, headache, body ache, mild rash, and fatigue. So it's, a little more than your COVID side effects. So that's what they are, but it's not deadly, right? And I think, and it's no myocarditis as far as I know. So if you need, not many people are vaccinated, but certain people are. The military are still vaccinated and so forth against smallpox. So I think it's okay. Know of an 8-year-old boy who has been infected with COVID 3 times. Last time he ended up in the hospital with multiple blood clots in the brain. What happened to him immunologically? So I can't speculate. Well, but I do know that with adenovirus vectors, the idea is that one of the clotting factors is binding to the vector, and it's stimulating the production of antibodies against the clotting factor. And those then trigger clotting. And so maybe something similar is happening here. Maybe the boy made antibodies that triggered clotting. Just don't know what's going on in this particular situation. We'd have to have more information. As far as I know, only single-cell organisms live in the Dead Sea. Yes, they would be resistant to the high salt for sure. Yeah. Hmm. Board the windows with solar panels, huh? That's an idea. But then you can't look out. I can definitely and happily report that the tone of today's show and the questions are far less serious than past weeks and months. The stress level of COVID has improved. I think that's good, right? I'm glad that we had over 700 people here still. That's great. People who are still curious. It's a community. You want to have discussions with other people. And you still have questions, and many of them have been not COVID, which is great, because I can teach you all of virology. We can teach you all of virology for sure. Dennis, my visual advisor, you don't need a solar panel, just charge on an outlet. Is that correct, Ian? Getting over Omicron, how long does the cough last? Well, I mean, it can last days or weeks. It can persist, unfortunately, which is not what you want to hear, but that's the range that we see. How do we work out which scientists to listen to? Michael Osterholm seemed legit, but you're not a fan. No, I'm not a fan because I think he overstates a lot of things and misinterprets a lot of things. How can the general public work out who's who? Very difficult. Listen to us. I mean, why not, right? We say what you need to know, so listen to us. And if Osterholm says something, ask me about it. But many times during this outbreak, he said okay things. He says get vaccinated. That's all great. But he's also talking about, variants wreaking havoc, and it's just not right. So, he's a pundit, I would say. They go to him all the time, and he loves it, by the way. Hey, let me tell you something. Before the pandemic, I had a podcast since 2008, and I invited him on. He said no, and now he has his own bloody podcast because he realizes how important it is. That just pisses me off. But that's not why I disagree with him sometimes. Yeah, he's on a lot of committees. He's well respected. It's fine, but he's not right all the time. Neither am I, but I'm not a pundit. One of your first ancient twivs was on rabies. Yeah, one of the early ones. But I'd like to revisit for sure with an expert. I know some, and we can try and get them on. Thank you, Real Qualia. Since the half-life of IgG is a month and mRNA vaccines rely on avidity, not affinity, at least in Omicron, what is the point in boosting? Because, well, no, it's not correct that the mRNA vaccines rely on avidity. Avidity is IgM. Affinity is IgG, and that's what boosting does. It causes more affinity maturation, somatic hypermutation of the antibody genes, and selecting for higher affinity antibodies. That's what the boost does. And let me remind you, this is because the first two doses are too close, in my opinion, and that of many others, and it's supported by the science. And the boost fixes that. It's basically giving you a second dose at the right distance from the first one. The first one was too close. The second one was too close to the first one to allow proper maturation of the immune response. As long as the causes for and remedies for long COVID are not well understood and still occurs, I don't think viewing COVID-19 as just a flu will stick. Well, don't you think that there are long-term effects from influenza that we don't really hear about very much? There certainly are. So yes, it will be because of that. How close have we come to understanding post-polio? No idea. There's certainly no virus present, but the best you can do is say the polio virus infection destroyed a number of neurons. As you age, you lose more, and you don't have a backup. And that's a simple explanation. But whether there are antibodies involved or what, nobody knows. There's no animal model, and interest in the disease is waning. So I don't think we're ever going to get there. Where can we or our doctor find reliable information about incidents and treatments of specific types of post-vaccine adverse events? I don't know what types you're talking about, like myocarditis with mRNA vaccines, blood clots with adenovirus vaccines. Those are, they both can be treated very well, the myocarditis in particular. Now, unfortunately, the blood clots, they happen so quickly they can kill some people. But those are standard treatments, and Daniel Griffin would be the person to ask about that. If you want information, email him, daniel@microbe.tv. And now we are saying goodbye to Amy. That's cool. Look at this. I'm like 75% through the questions. That's amazing. I don't have a cozy fire. No, I don't. It's off. See? I don't have it on. I would make noise and but I understand it would be cool to do that. Can you elaborate on the status of Amy's grants? I did that last time. And so basically she needs to get funding to remain at Columbia. But we have hopes that we have a grant in that will be funded. We don't know. But she also has some irons in the fire for a job, so let's see how that turns out. She might be okay. Did Amy say we all need to die off before COVID becomes epidemic? She said 50 years. Endemic. No, it's already endemic. What did she say about... she thought about it when people just treat it as another virus. She thinks 50 years. And, the remark about long COVID is a good point, because as long as you have long COVID, it may be hard to just look at it as another virus, right? But I think that 5 to 10 years is in the cards, because this is rapidly... Look at the situation we are in now in countries where we vaccinated. Cases are dropping. And I think we will have a few more rounds of outbreaks, but I think people are getting used to it, right? The tone tonight is very calm. So, it's the press that likes to froth up the excitement, but I think it can happen sooner rather than later. Is long COVID overblown? No, it's not. It's a real thing with people who are infected without being vaccinated. But vaccination, especially boosting, really brings it down. It's not zero, but it's very low. And so I don't think you should worry about it. Am I a vegetarian? I am not, but I do try to avoid meat as much as possible for health reasons, but also I feel badly for animals being slaughtered for. I'd rather eat pasta and salads and all kinds of vegetables and desserts without consuming animals. Got a natural infection as a booster. What's the ideal spacing to get a booster? So you got vaccinated and then you were infected. I would wait 6 months. But I think you're already okay. You don't need a booster. But Amy did indicate earlier that the current suggestion is to get a booster. So I would wait 6 months. It's a good spacing. When a person with 3 mRNA shots has an infection, what does that mean? Well, you're getting a boost. And this is going to happen now for the rest of your life, right? You're going to get infected every few years probably with this virus. It's going to give you a boost. And you're going to have more protection as a consequence. And the infections will all be mild. That's the pattern in the vast majority of people, not in everyone. Thank you, Datebio, for your contribution. What are the odds that the Pfizer under 5 doesn't produce good results after a third dose? I don't know about odds. I'm not sure that it's going to fail. I have optimism that it's going to do well. But if it doesn't, there is There are other vaccines that are going to be tested, which might work, and you have to keep them masked and distance in the meanwhile, and make sure the adults around are vaccinated. You guys are talking about my diet. Just ask me. I do eat cheese. I'm not even a vegetarian. I'm not. So Andrew says, not if he eats vongole. Okay, but I'm not. I never said I was. I do eat. I eat fish, right? But I worry. It bothers me even to eat fish because they're beautiful animals, right? I don't even like to kill spiders and bugs because they're cool. They're very cool. Why should I kill them? All right. And you can get plant-based cheeses. Yeah, it's fine. But I don't have a problem with milk. Okay? At least we don't need to get into this debate. Eventually those cows get killed for meat, but I'm drinking their milk. And I don't drink much milk at all, but I just like the cheese, man. Cheese is awesome. Thank you, Sherry, for your contribution. People are here talking about food and other stuff, and it's great. And then people say, why are you wandering off topic? Give me a break. I worry my memory T's and B's won't come to the rescue. Why not? Why do you worry? Like, what's the basis for your worry? Do you have any data on it? Or is it just a human worry? Your T cells are great. They protect you against severe disease. Maybe you in particular? Sure, it could be. But that's just paranoia. Chill out. You'll be okay. The data say that most people are going to be okay. What makes making an RSV vaccine difficult compared to other? Well, part of the issue is that they tried making one in the 60s and it failed and it made disease worse, and kids died when they got infected after the vaccination. So people have been, first of all, trying to figure out what went wrong, and then, not wanting to duplicate that. So it took a long time. And now they're finally getting going again, and we have some interesting candidates. But the other issue is babies get preferentially infected in the first 6 months of life, and it's hard to immunize them. And so one approach is to immunize the mother, and then the antibodies transfer across the placenta. And so that's one vaccine that's in testing. But older people get RS as well. We've only recognized that recently, so that's another part of the equation. Where's T cells when you need them with rabies, when you have to rely on post-exposure? Well, most people are not vaccinated against rabies, right? Veterinarians and people who work in the forest and with animals, they're vaccinated, but most of us are not. And you get bitten, you're not vaccinated, so you get post-exposure. That's why. It has nothing to do with waning anything or contracting anything. Well-informed uncertainty is a hallmark of good science, for sure. Yeah. You have to say, I'm not sure because we don't have the data. I always say that I base my opinion on the data. If there are no data, if you say this virus is more transmissible, I say, show me the data. And then if you don't have it, then I say you can't make that conclusion. And that's what's happened throughout this pandemic. Sorry for asking this. No sorry. It's okay. But the young kids who've had shot 1 and then infected need shot 2. So the data say no. CDC will say yes, you should have 2 and an infection. But the data say that 1 shot plus an infection gives you a great antibody response. We are meeting in Madison. Yes, we're going to meet. We're going to have a meet-up in Madison. I will be there this summer, July. I can tell you exactly when. February, March, April, May, June, July. ASV starts Saturday, July 16th. So I will arrive then, and I will leave on Wednesday, the 20th. So one of those evenings we could meet up at one of those cool places in Madison. It would be fun to meet some of you who are on this stream or are listeners and so forth. It's excellent. 2:30 a.m. in Ireland. I'm so sorry. Sleep is calling. Go to bed. Thank you for coming. Is the lesser mortality in Australian rabbits due to the host response or a change in the virus? Well, these are European rabbits. And the Australian rabbits are resistant. But they couldn't hunt them, right? They were too evasive. So the European rabbits were huntable. I don't know why. So that's why they imported them. But as the European rabbits changed, yes, they sequenced the rabbit genome, they sequenced the virus genome, and they identified changes in both. And we did a TWIV on that, actually, which you could find for sure. Is anyone studying a possible similarity between ME/CFS and long COVID? For sure. There are, because there are similarities and differences. I know Ian Lipkin is a big studier of ME/CFS and is also interested in long COVID. So, and others are doing that as well. Absolutely. If someone has long COVID symptoms, but the exposure occurred early in 2020 before antigen PCR testing was available, is there any way to be definitively diagnosed? Nucleocapsid antibody test was negative. That's the only way, is an antibody test. If you've not been vaccinated and you had antibodies, against the virus, that would be the answer. I would get another test with a different assay, just in case the test was faulty. But after 2 negatives, then they weren't infected. They have these symptoms from something else. Thank you, Kev. Do common colds spread better than flu? Depends on what flu strain or variant we are talking about, right? Because some of them are more common cold-like than others. But common colds do spread very well because they're largely innocuous, and people tend to go to work and do whatever they do while they're infected, sniffling, and they spread it, right? And so that's less common with flu, which tends to knock you out, but there can be infections with flu that are milder. So there's an overlap. You know, as I say many times, nothing is black and white, right? Thank you, Samster, for your contribution. Restating part of my question, is the original virus without mutation introduced in Australia virulent to the rabbits in Australia, not the virus that evolved and became less virulent? I don't know about Australian rabbits. I believe they're already resistant to the virus. That's my recollection, because I teach this every year. And it's the European rabbit that is killed by the virus. Okay? Do you believe there's any way to cover patient 0? No. If not, what do you think is the chance of this originating in a vaccinated versus unvaccinated individual? I think the highest chance is that it originated in an immunocompromised patient, like an AIDS patient, where vaccination wouldn't matter because they're untreated and they're immunosuppressed. And there is an interesting preprint out of South Africa of a young lady who went untreated with AIDS for 9 months, and she got COVID, and she evolved virus with a lot of changes similar to what we see in Omicron. Not all of them by any means, but kind of. And then they gave her antivirals, cured, or let's say tamped down her HIV virus load. Her immunity returned, and then that cleared the COVID from her, the SARS-CoV-2 from her. So I think that's the most likely origin of Omicron in an immunosuppressed person with an abnormal immune response, allowing the virus to reproduce for many, many months and many changes to accumulate. If you lose your sense of taste and smell, does that mean the virus entered the CNS and affected a cranial nerve? No, it doesn't at all. In fact, we know, and the studies show this, that the virus goes into your olfactory epithelium And they're nerve endings, right, which sense chemicals, odorants, and the nerves transmit the signal into your olfactory bulb in your brain. Those nerve endings consist of the nerves and support cells called sustentacular cells. Those are the cells that are infected by the virus, the sustentacular cells, and that it destroys them, and that impinges on olfaction. The virus does not get in the brain and reproduce there, and has no effect in the brain on olfaction. And my cat name is Luna. She had a URI a while ago. Don't know if it was corona. I'm not sure they are. I haven't followed this, whether they're vaccinating animals. I'm not aware of that. They're certainly vaccinating zoo animals, right? Could naturally occurring pseudouridine explain latent PCR positives? Why might some produce this peptide more often? I'm not sure. So, yeah, of course, there's naturally occurring pseudouridine in like tRNA, right? But you're saying it's incorporated into the viral genome and it lasts a long time? I'm not sure that it's available for general incorporation. That's a good question. I don't know. There's a lot of talk on the news about a fourth shot. If the vaccine is not changed, is it worth taking it? No, you don't need a fourth shot. Fourth shot is useless. This is BS pushed by some public health people, and the press is just picking it up because they need to fill the space. You don't need a fourth dose. I am a flight attendant and very concerned about the mask mandate being lifted in a few weeks. You've probably already spoken with Vanity, who is currently in the air, about the mask mandate being lifted. Well, do you have to lift it on yours? Can't you wear a mask? I think so. I go into a restaurant and I'm not masked because I'm eating. The wait staff is largely masked because they're there all day and they're contacting a lot of people. I think it makes perfect sense for them to be masked, especially if there's a lot of virus circulating. So you could just mask up. But I think if you're triple vaxxed, you're good. You're going to, at worst, have a mild infection. So if they require you not to mask, I think you're okay. Thoughts on COVID and genetics, specifically OAS1 with the risk allele that affects the P46 isoform and RNA cell is inefficient. So, yeah, OAS is a host defense protein, and it is particularly important for coronaviruses, as Susan Weiss has shown. I'm not aware. Are there studies that implicated for SARS-CoV-2? But that's the kind of host genetics that could cause someone to get severe COVID and not others. A good RNA cell versus a not so efficient one. And genome-wide association studies, sequencing genomes of people with severe and mild disease is the way to get at it. What human challenge study would you like to see? I don't want to see any, frankly. I think they're unethical. And you cannot do a challenge study where the risk of long COVID exists, or even of death. I don't care that they had 34 people and nothing happened. What about the next 34? When someone dies, what are they going to do? When someone gets long COVID, how are they going to feel? That's unethical. I'm sorry. Do no harm is the mantra, right? Anyway, I would like to see vaccinated and unvaccinated people challenged and measure infectious virus shedding, level, and kinetics, so we can get around this nonsense that vaccinated people transmit as well as non-vaccinated. There's just no evidence for that. That's what I would love to see. And Shari, thank you for your contribution. And I just chatted about your question previously. The media keeps talking about waning immunity after booster. Aren't we still protected? Yes. Memory B and T cells protect us. The media doesn't know their basic Immunology. It's too bad, right? I wonder if vongole would grow in Australian waters. They don't grow in the U.S., right? That would be interesting. They're very tasty. Very tasty. Oh, so here we go. Vanity says to Allie, who's the other flight attendant, I think they may extend the mandate again because the vaccine isn't available for those under 5. Date is coming up, though. Yeah, that would make sense because a lot of less than 5-year-olds travel, so you should protect them. Yep, agreed. Could you do an episode about SARS-CoV-2 and pet animals, natural hosts, etc.? Well, there isn't that much information. And whenever we do an animal episode, we try and summarize it at the beginning. But when we have more data, we will do it for sure. Absolutely. Goodbye, Andrew. Thanks for coming. See you next week. Nine minutes more, folks. Don't mean to keep you all up, but I do want to hit 10:30. It looks like a lot of people are leaving. Hey, I do have a pen fetish, folks. I don't know if you know that. I haven't talked about it, but I love pens. In the old days, I used to buy a lot of them. Got a cool one right here. This is a Rotring. It's metal. It's a pen, right? Rotring 600. You can't see it. It's out of focus. I love this. It's got such weight. It's a nice clicky ballpoint. Pens. Jetpens.com was my pick years ago. What a great website. Thank you, Marie, for your contribution. Really appreciate it. Do you and Amy blog? I've written virology.ws for over 12 years. And up until recently, I was blogging once a week. I would release a post on Thursday night. And I've felt full and short. I've been so swamped in the last couple of weeks that I can't write anymore. I'm editing podcasts, but I hope to get back to it. But virology.ws, I have other people writing, David Tuller about ME/CFS, Trudy Ray about viruses in general, and I have a lot of posts there over the years. And Amy has a blog, enterovirus.net, but she doesn't post all that much, but she is a little busy. So that's to be understood. Two children under the age of 5. Are we waiting at April? Looking at April, most likely, probably most likely. And thank you. There's the there's the blog. Thank you. I heard somebody claim the dangers of vaccination for youngsters are greater than COVID. Am I correct to say, Daniel, when you disagree? You are correct. It's just not true. A lot of kids have been dying of COVID in the last couple of months, more so than earlier. 700, 800 kids. If that's your kid, would you say the vaccine is more dangerous, especially when their side effects are no different from those in adults? So that's just wrong, yeah. What's my definition of a T-cell epitope? T-cell epitope is the protein sequence that is recognized by the T-cell receptor when the peptide is presented to the T-cell receptor in an MHC molecule on the cell surface. So the viral proteins are chopped up by the proteasome. They're presented on the MHC on the cell surface, and then the T-cell receptor recognizes it. That's the T-cell epitope. And they vary much less in spike than antibody epitopes. All right, so here's the thing, Sally. The study you saw reported in the media about protection decreasing with increasing time looked at emergency department visits and hospitalization. This is flawed because that's not direct. It's an indirect measure of the protection. For example, hospitalization, you could go to the hospital for something else, and if you test positive for COVID, it's written down as a COVID hospitalization. So I don't believe any of these hospitalization metrics. I just want to know severe disease and death as measured by symptoms, and they typically don't do that. And you have to also understand that all populations are different. If they do these studies in 80-plus-year-olds, yeah, they have comorbidities. They're older. They have weaker immune systems. It's not going to be the same in everyone. So to be very skeptical about those. I read in the news that too many boosters could use up all your antibodies. Oh, that's BS. That's just something on a shingle, man. No, it doesn't use up your antibodies. Boosting, you don't want to have 10, 15 boosts. It's not necessary to begin with. And I'm not sure of the harm anyway. Maybe you might generate some autoantibodies because you're pushing your body to do somatic hypermutation so much that maybe they would pump out antibodies that react with you. But we don't need to use that many boosts. Are the rapid tests accurate? Yes. So they depends on which one because they're all different. But there was a wonderful study in, I can't remember, it was on one of Daniel Griffin's recent clinical reports, which looked at the comparison of rapid with PCR in different times after onset of infection. And it's quite good. The accuracy showing that you're infectious is quite good. They compared it to infectious virus. Doreen, thank you for your contribution. I'm glad you like the pods. And yeah, I love coming here and talking with you guys. It's a lot of fun to interact. It's why I like to teach undergraduates. Right? They're not all scientists. They're from all walks of life. It's a lot of fun to get them excited, and I love doing that. And it's great. I've discovered the live stream in the past couple of years, so I'm going to keep doing it. Want to figure out excuses to do it more, right? Thank you, Rima, for your contribution. Really appreciate it. And let me get through the rest of the contributions. We're almost at the bottom. It's not bad. We did better this week than most others. Thank you, Mystery Fine, for your contribution to the incubator. Can we pass 600 likes? We're at 548. It's Nathan's idea. Thank you, Nathan. The questions are all great. And if you didn't get your question answered, bring it back next week, because we will be back next week. Codswallop. Thanks for the passing reference to bat fungi. It's a passing reference. Okay, everybody. Unless there are any other pressing issues, let me see here. I think there is a This Week in Pens. There is a podcast on pens and papers and inks and so forth, which I listened to a bit, but I didn't really like it. From what I understand, you need a new flu vaccine after 4x drop in antibodies. Yeah, because we know that, and it's not just antibodies, but it's hemagglutination inhibition antibodies, because they know that correlates with an increase in disease. We don't know that for SARS-CoV-2. And that is the reason why we can't say a fourfold drop is enough. Because, in fact, a fourfold drop in neutralization, Omicron is much larger, but you're still protected against severe disease. So we don't know the correlative protection. That is the problem. All right, everybody. I'm going to end 2 minutes early. Thanks for coming. I just see a new contribution here. Linda, thank you so much for your participation. There we are. Linda, thank you for your contribution to science education. Thanks to all the mods tonight. Vanity, Les, Steph, Tom, and Frank. We really appreciate your participation. Thanks all of you for coming and for your great questions. And bring them back next week. In the meantime, listen to TWIV. Thanks, everybody. Stay safe. Good night.